one. But anyhow, source instructions. थोड़ा
Recording in progress.
तो स्लाइड प्रेजेंटेशन किसने करा? बंद रहा है क्या?
Dr. Das sir, with your permission, may we start?
Please go ahead.
Namaskar and good afternoon to all of you. On behalf of Honorable President, Founder President, I am delighted to welcome Dr. Rajesh Khanna, who is Director of Pain Research Center at New York University, USA. To this campus, Noida, let me share with you, this is one of the 11 campuses in this country and one of the 24 or 25 campuses world over. Why I'm taking liberty of saying 24, 25? Because we are growing so fast that, today if we are 24, maybe after some time, some few days, we may be 25. And Founder President Dr. Ashok K. Chauhan sir has sent greetings to you because of some other commitment. He is not able to come here physically, but certainly he has assured us that he will be joining in between the program. We at MIT are thankful to you for having consented and to be with us this afternoon to share your research and your readings and your thoughts with our faculty. I know that you are very busy. This morning you had given a talk in All India Institute of Medical Sciences, New Delhi. A few words about MIT. Today MIT stands very tall and it's the largest university in private sector in this country. And we are proud to have more than 2 lakh students and who are guided and taught by more than 10,000 faculty. We have 28 schools, means the MIT provides education right from primary school to postdoctoral fellowship. And we have about more than 120,000 alumni spread throughout the globe. And this university offers various courses and they span right from agriculture sciences, environmental sciences, neurosciences, defense technologies, robotics, and artificial intelligence. So the canvas of research is quite high and also the courses offered to the students. And we also boast of having filed 1,860 patents. And the average H index of the faculty is 112 at the moment. And the Honorable Founder President has given the target that it should cross 150. We have about 400 projects running, which are funded by various funding agencies. Dr. Khanna, your talk on pain draws special attention this afternoon because the Honorable Founder President has shown keen interest, and he has envisioned and he envisaged that MIT wants to start the Center of Pain Research. And your presence in MIT campus shall open new chapter and a new starting, and your presence will not make us more learned on this topic, but also pave the way for collaboration, which we would like to nurture to strengthen the pain research between your university and MIT. I was talking to Dr. Rajesh Khanna that he went to AIMS and he was coming. When he was sitting in the waiting area, he couldn't believe that he is in India. He says the infrastructure and this university campus is so impressive that he said that it looks that I am in US. And when I was replying, I said, yes, this is the vision of the Honorable Founder President, that who has created this. And this university is in the transition. I think I said, and I again repeat, that tomorrow belongs to us. The tomorrow belongs to MIT. And once again, I thank you and I welcome you to this campus. And we all are eagerly waiting to share or to listen to you. And thank you very much. No, now I think Dr. Das will. Yeah. Sure.
Okay, but then this slide will not be visible.
No, we can
Okay, sit there. Okay. So, good afternoon, one and all. And at the outset, I welcome Dr. Rajesh Khanna from the New York University and the director of the Penn Research Center. And it's a great occasion today. I, on my own behalf and on behalf of our Honorable Founder President and our all colleagues in the MIT, the faculty colleagues and all others and students and others. So I welcome you to MIT University, Uttar Pradesh. It is called Uttar Pradesh and situated in Noida. And this is the main campus or the original campus, the oldest campus. And now we have almost a dozen campus. I will just briefly, maybe 10 minutes time, I'll just complete that so that you have some impressions and you are first time visiting India. And of course, the MIT University first time, you had been to one of the top medical institutes of the country, All India Institute of Medical Sciences. And this is one of the top private university. You happens to be here. So let us see what we have been doing it here. And so then later we will discuss other things and I will listen to you. But one important thing, those who have gone through the CV of Dr. Rajesh, so he is an excellent scientist at at bar, that you will see the publications. There are all top journals, and every second paper in JBC. Those who are in hardcore science, they know that what is the value of the publication, one publication in JBC, particularly in all over the world and India. If anybody publishes, everybody comes to know, oh, that it is a JBC publications. So, and he works on an excellent field of the pain, and which is we are all aware of, and particularly Professor, and I welcome other people, all the people, but on the dais, Professor Singh, Professor Sharma, and Professor Professor Malik. So B.N. Malik, he is here, and he is our head of the director of neuroscience. He was the professor of JNU. So he is welcome, Professor Malik, here in this meeting, because you will be more closer to your uh, the pain. Because as I just before coming over here, I was just briefly telling you, it happened so that myself and my colleague, Professor Seema Bhatnagar, we just submitted a project. Though it didn't get through, there are some technical problem, but rather than scientific problem, NTPT. So that was on the for the neuropathic pain in cancer. So that's a project on that. So that may be on during discussion. We'll discuss that. But briefly, I will just go through. So in the neurology and the physiology has the intimate relations with the pain. So, and if you can do something to pain relief and pain management, that will be a great contribution to the humanity and to our society. So, this is Amit University is what is called as the we nurture the talent. So, I am rather say I borrowed the slide from Dr. W. Selvamurthy. He is not here. He is the president of the Amity Science, Amity Science and Technology and Innovation Foundation, ASTIP. So, which the vice chancellor takes care of the academic affairs of the country, of the university. But this is a organization which is or the unit made by it is the brainchild of our none other than our honorable Honorable President Sir, who is a great visionary. And as I briefly told you, and many of the things, many of the new departments, like my own department, the Amity Institute of Molecular Medicine and Stem Cell Research, the nanomedicine, long back, it was many institutes have been created for the first time in the country here only. So like nanomedicine, people are doing a lot of nanotechnology, but nanomedicine is there is no other department except in Amity. So that is headed by Dr. Professor Tinku Basu is sitting there. So like that, there I'm not taking many of the names like the defense strategy, defense school that has been established, and robotics and many other things. I will be coming to that. So it is Amity is that it is told that it is said that we nurture the talent and we build the nation building through education and research. And this is the whole campus picture. You can look at it here. And this is a huge campus, and it is now added with the another new campus. In that, with this, it's called Dell campus, where our colleagues, the AFSIA, we call as Amity Foundation for Science, Technology, and Innovation Alliance. So they are for India and abroad. They make the alliance. That's why it was Dr. A.K. Singh who took the initiative that he contacted you and he has brought you here, and we have several visitors time to time, including Nobel laureates and all. So this university is the founder is the persons who has built this education empire. What is his just first slide I am showing? What is his thought? He says we should be the knowledge superpower by 2030. And every day, every moment, he comes with a new thought. So we are always eager to learn from him, and he is really is a great visionary. And Amity's mission is the Amity's passion and Amity's junoon. So you know, so he was born in India here in Delhi, and then he went back to in childhood. He was for some time here. I don't know. He will share, and then he has since then he was abroad, and he is first time visiting rather India after 36 or some years. So he was in Canada, then to US. So he says the nation building through education, research, and philanthropy. inspire somebody, lead somebody. So no power in this world can stop such a person. So this is his philosophy and his word. And most important, I am showing his two sons here, and there are others who are educated from US or UK. So it's, Dr. Atul Chauhan, who is the chancellor of this university, the Noida MIT and other MIT University also. He is also president, is the Balbed, the Ritnan Balbed Education Foundations, is called RBEF. So, which all the education or the universities come under their in these foundations. And so he is a product of London School of Economics. And another, Dr. Asim Chauhan, he is also the product of Wharton's School of Business School and from US. And so he is the chancellor of the Haryana Amity AUR, Amity University, Haryana. And he is also happens to be the also the chancellor and I think of the university who looks after the university at New York, which is I think that is St. John's University, or that is in front of the sea. So that is near that big laboratory. That what is that laboratory? The famous lab, which is called the. I'm just forgetting. So this is a 175-acre the space and the built building, as well as the huge space there in the New York. So this the Long Island. So this is the one. So I think so. These are the what is I want to show that these are the highly educated persons are the in the management of building up these education groups. So this is what that it is not that they are all the highly educated people who are looking after or in the management, and that's why how to navigate and how to go ahead with the frontier lines. So they are very much aware of. So the Amity University, just briefly to tell you that this is the Noida campus, and then there is a Gurgaon, the Haryana Amity. So founder says that he wants to see each state having one Amity University. So we are at the moment 11 or 12. So like Greater Noida, Mumbai, Lucknow campus, and then Jaipur, Gwalior, Raipur. So there are other campuses in the abroad. There are 17 campuses abroad. So Amity University, Singapore, Amity Mauritius, the Amity New York. This is the campus, as I told, Long Island campus. The Amity Nairobi, Tashkent, London, and the Dubai campus. So there are at least more than 300 UG and PG courses. So there are, you name it, it is there. So both science, technology, and innovation, and health and allied sciences, and many other natural sciences, environment, and all. I'll not go into all these detail, but this university is ranking university, the NRIF. You must have heard. This is the national institutional ranking done by the UGC, and there is also NAC ranking, which is the NAC A plus, and the university is 22 in the NIRF ranking. Pharmacy is 17, management is 28, and engineering is 25. So they are also having the US ranking and the WASP and their QA ranking. They are all this is the university which has.
The Indian Intellectual Property Office of the Government of India has conferred the National Intellectual Property Award 2020 on Amity University for Top Indian Academic Institution for Patents and Commercialization, a reflection of Amity's commitment to research and innovation.
So every time, every day, every week, rather the patent goes high. So at this moment, I think it is 1860 patents. So every week there is at least 2, 3, 4, 5, or 10 is added. So it's a huge campus. So this is Also, it has also got the special appreciation award from the excellence in the award in the to the Amity University. So this is a called question. Question. So this is also a prestige to the Amity. So it is, in a nutshell, you have at any time point of time now more than 2 lakhs students, all the campuses together. And there are 10,000 faculty and scientists and 11 university campuses in India. And there are 17 campuses abroad and 150 institutions. And this is this campus, and there are 28 schools. So what was meant, said by Dr. Singh, that it is KG to PhD. You enter into KG and come out with PhD. So including postdoc also you can do it. But mostly in India, people prefer to go abroad. So this is the basic principle, and so they have Amity International Schools with very prestigious. They come many a times to the top positions in the country. So global higher education campuses in the London, that is the international campuses, global school campuses also, Dubai, Abu Dhabi, Singapore, and South Africa, and now Amsterdam. So there are many other schools abroad, and then there are at least 120,000 alumni are distributed throughout the world, and 300 plus courses are there already. I told the programs, and 30 plus centers for the excellence centers, and 1860 patents so far, and the 30 technologies are transferred, but many are in the process, in the way, and there are huge number of publications, more than 3,000. 30,000 publications, and they are in the from minimum 1 or 2 factor, two digits, even 3 digits publication means in 30 of publication, 30, 36 or 20. So a huge number of publications, including Nature Genetics and Lancet and many other high index journals, and the nanosciences and all, and neurosciences. And there are several books. I will not go all the details on this. There are important things that more than 300 projects. Some institutes have the, because there are huge number of Ramalinga Swami fellows who come for 5 years. They are selected from abroad, and they come with the DBT's fellowships. So they gave huge amount of the contingency and the fellowships. Their salaries also maintained till they find a permanent placement. So Amity has the highest number of the Ramalinga Swami or Ramanujam fellow or fellow in all campuses, and this campus having the more than 25 fellows, and some institutes has, like my institute has more than 12, and which is any of the single institute has the highest number. So this is because of the fact they are coming where there is a facility. They can do their work, because everywhere they have to go to the conclave and present their work. And it is only just 2 months before the conclave was there, and it was announced by the DVT that the maximum number of Ramalinga Swami fellows and Ramanujam fellows, they come from Amity University. So this is a great prestige. And there are more than 400 projects, research projects. So, so the huge number of projects here, and so that's why the research publications accordingly, and they are getting the projects because they are good publications and good research contributions, and patents and research contribution publications. So this is also a research promotion. There are several facilities are given here, sponsored projects. Then we have in-house research facilities, the state-of-the-art, the laboratory facilities, and they are still preparing. Many of the facilities are being built up. The Amity research concern, that's the connectivity. So it is the synergy of the brain, as our founder president says, the synergy of the brains for the betterment of the knowledge and innovations. So it is connecting, seamless connectivity to the stakeholders, industry, university, scientists, and society. And IPR protection cell, Directorate of Innovation and Technology Transfer, DSTA, sponsored MET, Technology Enabling Center. So those who are in TRL 3, 4, we give them some grants. We also mine around in the country, in the Delhi NCR, if there is any technology, and in the university first and then others, and we give them some grants and to make them a industry level ready. So TRL level 7 or 6, 7, so that they can be transferred to the industry, so that that comes to the use of the humanity or the society. Amity Center for Entrepreneurship Development and Amity University e-cells and all these. So Amity research, the senior researcher, the most important thing is that all the senior people, in India, you might be aware that after 60 or at best 62 or 65, different level of retirement. So most of the research institute, they get retired by 60. So they are 60 is nothing but they are young. I would say they are young. And from there, they are appointed here. They are selected and appointed here. They are coming with the excellent experience throughout their life, and those who are excellent, they are being appointed here. These are the proof. We see that a lot of senior people are here, so they mentor the youngsters, which are young faculty and all. That is the beauty and important things in this university. So this is, and there are several collaborations and honorary professors and visitors from Nobel laureates and many of the adjunct professors from USA, UK, and all these places. I have not go. So these are the senior people. You will see, you may not be aware of them because you are abroad for a long time, but they are very senior and they are either vice chancellor or the advisor, and they cannot mostly hold the regular positions, but they are like advisor or the senior scientist. So they guide the people or the junior faculty and scientists, so that how they are helpful. So and these are the young people, the Ramalinga Swami fellows. So there are several of them. They are a very young blood, and the Welcome Trust fellows. So there are all types of fellows are here, and the TC I have already explained, and DST supported Amity Innovation Incubator. There are more than I think several incubator, the 300 plus incubators are there, and there are international collaborations with Europe, USA, and Asia, other places, and the startup companies are there. More than 70s are startups by the students. So there are all. So I will not go all these details. And then the Amity Research Ecosystem, the Amity Science Technology Innovation Foundation, as Steve I told. The another is the ADCI, the Amity Directorate of Science and Innovations, and the one Dr. Singh has called for, this is the ASTIA, we call the Amity Foundation for Science, Technology and Innovation Alliance. And so these are facilities you will visit, you have the time, then you will see there are all modular labs and all state-of-the-lab facility, and all types of modern equipments are there. And there are several MOUs. MOUs have been built up with the national and international levels. You will find there are all topmost universities in the US, and then Hopkins, University Kent, and UK, and Purdue University, and then the University of Florida, Wisconsin, so Colorado, and other in the Australia and others. We have collaborations, and then national CSIR, ICMR, ICAR, DRDO, all these, and then different hospitals. We have a collaboration because we need the clinical materials. Without, particularly in cancer, neurological, and other research, if you want to do good research and the translation research, you have to have a clinical samples and clinical correlations. You cannot do everything on cell lines. So we have to depend on, and we have the great collaboration into that we have a provision. No other university in the country have that you can have it is a must that you will have the external co-guide from India and abroad, so that, for example, you have come, and if anybody wants to work in this line, and if you agree, then we urge that collaboration, that you would be the external co-guide. The students can go and work there for 2 months, 3 months, 4 months, 6 months, and they are the part and parcel of them, and they get the guidance of the senior people. So this is how it is done, and that's why these from hospitals and all, they become the external co-guide. So these are and there are discipline like computational biology, cancer biology, translation research, molecular medicine, genome research, stem cell research, neuroscience research, public health, click chemistry. And this is a new discipline, click chemistry. There is a very good department, drug designing, discovery, ocean, atmospheric science and technology. We have a lab in the Antarctica. And then the Indian system of medicine also, we are not far off. We didn't forget our origin. The Indian system of medicine, yoga, and the Ayurveda and homeopath, and the data science, nanotechnology, robotics, aerostrategy, cybersecurity, space science, spintronics, artificial intelligence, photonics, artificial intelligence, everywhere. So even it can be used in the pen research, and all in the medical care or the healthcare, and the post-harvest technology in agriculture, food and agriculture, bioenergy, and I think Dr. Kaushik is here perhaps somewhere. So these are there isn't the innovation. There are, I will not go all these details. So there are some one or two I will mention. Nano composites for the drug delivery and wound healing. So there are also the targeted delivery of the that we have used the triple conjugate, that you can have the curcumin which sensitize the cancer stem cells, and then you have the folate. That folate is the signature or the receptor is highly expressed in the dividing cells, cancer cells and cancer stem cells. So that can act as a ligating target, target, and that conjugated with the standard drugs. So generally, standard chemotherapy drugs and radiation cannot kill the cancer stem cell. They have the transporter proteins, so ABCG2. So that reflects out the cytotoxic drugs. So when you sensitize with the curcumin, and it binds to the folate in the cell receptor, and then got into by the vitamin uptake procedure, endosome formation, and then got into the cytosol and release in the pH, it will release the curcumin, and curcumin will sensitize the cells, and then the drug. For example, we have made the doxorubicin. It acts and it kills the cancer stem cells, because this is these cells which are responsible for the majority of the cancer relapse. So this is they are also the repurposing of the drugs, novel synergistic formulations, and herbal remedy for the psoriasis and many things. And there are a lot of work has been done in the COVID-19. So then in the neurology also, the L-DOPA, the novel compositions of the treatment of the Parkinson disease. We have our colleagues who have worked on it's called type 3 diabetes. It is the Parkinson disease or the Alzheimer disease or Parkinson disease, where they develop they have characterized, they have published in the high impact journal that is called type 3 diabetes. So and then it is the bandages for the immediate relief and pain and accelerated wound healing. So these are few of the works have been going on, and that has been patented. And there is a combination drugs for the treatment of the brain diabetes, which is called the type 3 diabetes. And so these are few of just to give a glance of the things. There is a Rutonic, very famous, and it has features a lot of patents, and as well as it's transferred to the technology to the several countries. So that is a growth promoter and abiotic stress tolerance, and it is from fungus, derived from fungus, and Professor Ajit Verma has developed this thing. And there is agrivoltaic, photovoltaic that can be used for the agriculture field. So that land can be used for the generation of the power as well as the electricity. And this is this has been accepted by the government of India. So I will not go much of the detail of these things. It will take a lot of times. So there are a lot of research universities that as I've told, this click chemistry, molecular medicine, spintronic, these are aspects, research clusters. So this has been also to synergy of the brains, that we connect with all the universities as well as the other institutes in the country through different, more than almost 50 areas of the research, including cancer biology, artificial intelligence. These are research clusters. So this like cancer clusters. So where we weekly or by the twice in a month we organize, or once in a month when it is convenient. So we organize the lectures even from the abroad and India, and that to connect with the subject specialist to and call the specialist to talk on that. So to have the connect with the brilliant brains. So what is that? Founder President says 4 Bs. That is the search for the brilliant and breast and brilliant, and what is another B? Brilliant brains. So this is the main principle to develop the new knowledge and out of the box thinking. So drug discovery, development, and biofuels, establishing a global network on the MIT Global Research Network in the novel viruses, and MIT Global Research Network for neurospine or neurosciences. So research. So this is a clusters have been really built up. On COVID, I will not go into all the detail. There are several things have been developed during the COVID time. We are also, it has been directed to research in that field. So there are a lot of things, portable ventilator, herbal hand sanitizer, and there are several things have been developed and contributed to the this management of the COVID in India. And as you know that the ultimate was the vaccine was developed by the indigenous vaccine, and that has helped to manage our own system or the in our own way. And now that the other places are having high COVID, including the China now, but India, it was little higher, but now it is completely in a normal state, I would say. So thank you very much for this brief introduction. I have taken time. So
thank you, Dr. Das, for nice introduction and achievements about the MIT. Now, I think though we are running out of time, so may I request all the faculty members to just give brief introduction to Dr. Rajesh Khanna, so that he is aware of the audience. And I can start from myself. I am Dr. Ajay Kumar Singh. I am Senior Vice President in MIT Foundation of Innovation, Science, Technology Alliances. So I worked in DRDO for 37 years and retired from there as Director General Life Sciences. So here at FSTIA, we are just, working as an enabler in progressing the research, alliances, and the innovation in all the campuses of MIT, which I think.
Yeah. Okay, I have already given the introduction. I am chairman of the MIT Institute of Molecular Medicine and Stem Cell Research, but I also look after the Dean of Health and Allied Sciences, and then I'm also the chairman of the University Research Council, of course, as the vice president, and I was in whole life in ICMR. I retired as the director of the Cancer Institute of the Indian Council of Medical Research, and then I was director of the Biomedical Center in the Delhi University. And then after that, I retired, and then we joined here and created this new institute. And I'm also the officiating director of the MIT Institute of Biotechnology, which is huge institute. Is the biggest institute, having almost 100 faculty and some 2,000, 3,000 professors and all, and huge number of these centers within that institute. So this is my responsibility I'm doing here, but mainly on research, the cancer research. Thank you.
Thank you. Welcome once again, Dr. Khanna. My name is Neeraj. My background is I come from the Department of Science and Technology. I was mostly in the technology development domain and incubation entrepreneurship. I was secretary to the Technology Development Board, an autonomous body of the Government of India. We would be eager to listen to your talk, but just one or two points I want to say. From the management point of view, we always look for pain point. So you're coming in giving us a talk where we're looking for where is a pain point, and when there is no pain, there is no gain. So again, I would say that yes, this is very important for all of us, and this talk, I think, would give us very important aspect of how to do the pain management and what we have been experiencing there back in the New York. There are a lot of challenges that we face in India. And welcome once again from my side. Thank you very much.
Hello, good evening, Dr. Khanna. I'm Viren Malik. I come from a department here called Neurosciences, and a few of my colleagues and students are around to listen to you. That's it. Thank you. Thank you.
Thank you, sir. I am Dr. Jaswant Sethi. I just take care of Amity Institute of Health Allied Sciences. I'm the director for that. I have 3 departments: physiotherapy, occupational therapy, and physiology and allied sciences. I myself is a sports injury man. I'm super specialized in clinical ergonomics and sports medicine. Thank you, sir.
Good afternoon. My name is.
Because otherwise we will not have time.
Good afternoon. My name is Gopal Bhushan. I serve as Deputy Director General and Director of Science and Innovation. Thank you.
Thank you. Yeah, this is Dr. Biswas here. Yeah, I'm originally from JNU, where the Mullik sir was there, PhD. Then I was in Vanderbilt Medical Center in Nashville. I start my stem cell research, and currently I'm with BC Das sir in the same unit, Amity Institute of Molecular Medicine and Stem Cell Research. And my expertise in the liver cancer is working good for me. Even set up a new center for liver research with a collaboration with Dr. Monga, Pittsburgh. Anyway, I'm doing something. Thank you.
Hello, my name is Dr. Advisha Bhattacharya. I'm a Ramanujam Fellow in the Institute of Amity Institute of Neurosciences. Currently, I'm working on the effects of retinal degeneration on cortical information processing. Thank you.
Hi, I'm Dr. Malika Chatterjee. I'm again from the Institute of Neurosciences, and my background is neurodevelopmental biology with a focus on specific neurodevelopmental disorders like autism spectrum disorder.
Hello, sir. So I am Dr. Vijay Kumar, working as an assistant professor in neuroscience department, and my research work focuses largely on structural biology of neurodegeneration.
Good afternoon, sir. My name is Dr. Kanish Loach. I am from Amity Institute of Pharmacy, and my work focuses on neurodevelopmental disorders, addiction biology, and stroke injury.
Hello, sir. I'm Dr. Sudeshna Mukherjee from Department of Physiology and Allied Sciences under Amity Institute of Health and Allied Sciences.
Hello, sir. I am Dr. Sandhya, working at Department of Physiology at Amity Institute of Health and Allied Sciences. I have done my PhD and postdoc from University of Heidelberg, Germany, and my area of interest is in cancer biology, mainly looking for the therapeutic target and how to use them to design diagnostic biomarkers and prognostic markers.
Good afternoon, sir. I'm Dr. Rachna Mehta. I'm also from Department of Neurosciences, and at present, I'm working in behavior neurobiology, particularly sleep neurobiology and neurodegeneration. My expertise is in neurochemistry. Thank you.
Hello. This is Professor Tinku Basu. I am heading nanomedicine department. Our focus is mostly nano-based diagnostic devices. For example, recently we have developed antibody detection of COVID-19. And apart from that, we also work drug delivery for few neurodegeneration diseases. That's all.
Good afternoon, sir. I'm Professor Dr. Seema Bhatnagar. I'm Assistant Director at Amity Institute of Biotechnology. And basically, I'm a medicinal chemist and I work on breast cancer. So my work on the estrogen receptors led me to explore the ER beta agonists for pain. And that is the project that Sir was also mentioning about. So we want to move ahead in that direction also. So we look forward to some collaborations here. Thank you.
Good afternoon. I'm Dr. Pallavi Agrawal. I'm a Ramalinga Swamy Fellow at Amity Institute of Molecular Medicine Stem Cell Research. And our interest is in cancer biology, more specifically understanding epigenetic events and looking for therapeutic implications of epigenetics in breast and ovarian cancer. Thank you.
Good afternoon. I'm Bhupesh Sharma, and I'm from Department of Pharmacology, MIT Institute of Pharmacy. My area of research is in brain, specifically the ASD, ADHD, and dementia. We are working.
Okay, thank you. Thank you very much. I think we are running out of time. Okay.
Yeah. There are others.
Hello, Dr. Rakesh. Welcome to Amity. I'm Dr. Shazina. I'm a clinician by training and I have my specialization in public health genomics and clinical genetics. And I was part of the UPMC team with Dr. Joseph Glorioso, who also works on pain therapy. So I welcome you to Amity. Thank you.
Okay, thank you. I'll just take 1 minute just to introduce Dr. Rajesh Khanna. I think I'll be as brief as possible. Dr. Rajesh Khanna, who is PhD and an internationally known expert in pain research, and is currently director of Pain Research Center at New York University, USA, from 1st January 2022. Prior to joining New York University, Dr. Khanna was the associate director of the University of Arizona Health Sciences Comprehensive Pain and Addiction Center. He wears so many hats. If you really see, he is also professor of pharmacology in University of Arizona. He's also the vice dean of research faculty. So I'm not going into the details of all that. But the director of the Pain Research Center at New York University, his main aim and his main pursuit is to take the initiative in the pain research. Because as you may be knowing, that opioids consumption was one of the big issues in USA, because that led to the addiction and to many deaths. So, sir, this is a very important area. And in India also, this pain research is drawing attention day by day as we are developing, because the urbanization, the per capita income is increasing, the lot of stress, and so forth. Dr. Khanna is also credited with developing a new molecule for pain management, which he says designed and dubbed as 194. I think during his discussion, we will come to know more about it, which is a sodium ion channel blocker. And Dr. Khanna has got a huge grant from NIH to the tune of about 8 million. And he also gets grant from the private people, private ventures, and industry. He is founder of 4 companies. One is Regulonus and other 3 companies. He did his BSc in toxicology, MSc in pharmacology from Toronto, and Dr. Khanna got his doctorate in physiology from University of Toronto and completed his postdoctorate in the Department of Physiology and Cellular and Molecular Neurosciences at the University of California, Los Angeles. And he has held many faculty positions. I think I will avoid speaking all. Has about 175 publications to his credit with H-index of 46. And he has many awards, long list. If you see with the word size of 10, the one full page. Reviewers, member of so many science academies and scientific achievements. I am just leaving all of them. And he has guided many PhDs and trained about 16 postdoctoral fellows. Sir, we are eagerly waiting to listen to you, and the floor is yours. Dr. Rajesh Khanna.
Just to add one word. So I have seen his biodata, and that he works on the ion channel, this calcium channel, and the regulatory proteins that involved in the pain. But one thing I would say, in the movie world, Rajesh Khanna. He's a superhero, but this Rajesh Khanna is the real superhero in the pain research. So, thank you.
Well, namaste and thank you very much. That was such a generous welcome. I've never had an occasion to be at a meeting like this. I must congratulate you, your leadership, the president. You have set up an amazing institute. I will say that one thing is lacking: pain. It's absolutely lacking. I know pockets of pain were mentioned, but you need a pain institute. Absolutely. I think we're at the last of my slides. Can we forward this to the front? First. Thank you. Really an honor. This is a phenomenal institute, and I look forward to discussions and to fostering some collaborations with Amity University. It's really a pleasure to be here. Thank you, Dr. Singh. Thank you so much for the invitation. I really appreciate it. So I'll just give you a 10,000-foot view of my vision for the Pain Center and what kind of things we're exploring at NYU, where I just entered. And I sort of made it a catch-all in terms of what opportunities are we pursuing in the context of pain. I'll give you some vignettes, some examples of how we've been doing it. So we all know pain is a huge problem. It's not just an American problem, it's a global problem. It's an Indian problem. As you said, Dr. Singh, that urbanization is happening here. People are getting more and more affluent. Education is much more, it's coming through more and more. I think this is a critical factor in the growth of India, but also realizing that we have to figure out how to solve some of these problems for pain. Globally, this is a massive, massive problem. And it's very complicated by the fact that pain is multifactorial. It's a disease, it's a collection of diseases. It's not just one single entity. And that's wherein lies the challenge of how we solve pain. Just to give you some ideas of numbers, chronic pain roughly affects a third of the US population, which is about 100 million, which is not a lot compared to the Indian population. I read this morning that about 20 to 25%, depending on what study you look at, of the Indian population suffers from chronic pain. That is a massive number. Massive. Think about that. Massive number with the population of India the way it is. And a lot of these folks, pain management is very difficult. If you have a little minor aches and pain, Tylenol, things like that work very well. But it's the chronic pain that I'm talking about. This is where you really have very few options. And opioids have been much maligned and much used and overused and abused, particularly in the US. I'm not sure how that situation is in India, but in the US, this is a big problem. This is cartooned here. You can see the impact of the opioid epidemic was on everyone's mind, except, for the last 3-4 years, you had this emergence of the COVID-19 pandemic. But nevertheless, this is the big problem, and it's only growing. The graphs on the right are not the Republicans versus the Democrats. It's a map of the US over time, showing you the density of emergence of opioid use and abuse and deaths. So a lot of people are dying from abuse every day. And this is not just from the perspective of chronic pain, but it's also from, like, for example, dental pain. In being in a college of dentistry where the institute started, the Pain Research Center started, 1/4 of all your prescriptions for opioids are coming from whom? The dentists. You go to a root canal, you get a root canal, and they give you 30 to 40 pills to manage with oxypills, morphine, oxypill, just for a root canal. That's insane. These pills are then sold on the street for $20 to $30 a piece. So you can see how this has really kind of exploded and is now the gateway. Opioids, morphine, are the gateway drug for crack, heroin, and this is really getting out of hand. And for the younger population, about 8 to 10% of the younger population, all of their drugs are coming from dentists. So there has to be a lot of education behind this as well. So when I was living in Arizona in the warm climate, and was recruited to NYU because you can see a smattering of centers across the US in yellow. But strangely enough, much like here, there was no center in New York. So this is a great opportunity to set something that is very important in the infrastructure of not just New York, but entirely in the US. So this is the center, which is right smack in the middle of Manhattan. It's this beautiful building here, overlooks the East River and the Empire State Building on the right. And it's a gorgeous center. I just started it. We have buy-in from, as you can see, a lot of the different stakeholders, people coming in from chemistry, from nursing, from engineering, from nanotechnology, anesthesiology, of course. So we were trying to get in people from all facets of the university across NYU to build a very interdisciplinary center. And so these are some of the faculty that I've already hired. And this one right here. Thank you so much. So these are some of the faculty that have been hired that come from different disciplines. We have crystallographers. We have people working on nanomedicines, looking at nanoparticle-based drug delivery mechanisms. We have our physician scientist, who's also the dean, who is looking at oral cancer biology and oral cancer-related pain, a lot of tumor-based work, as well as younger folks who are working on receptor trafficking mechanisms. We have physician scientists working on irritable bowel syndrome and related disorders, which is apparently very huge in the New York area. So this is really the vision that's been set up, and we are now I'm now at a stage where looking to expand, hire new faculty, and grow the center so that we can occupy the entire building. So let me switch gears a little bit and tell you a little bit about the lab's goals and research. As I told you, pain is a constellation of diseases. So going at it in just one way is not going to be the best approach. I think we have to come at it in a multitude of ways, right? So I'll give you some examples here, and I'll tell you a little bit of each of the story, and then one story in a little bit of detail. Okay, so we can come out at it different ways. So much like in India, you rely on Ayurvedic medicine, natural products. Living in the west part of the US, in Arizona, I came across natural product chemists, natural product agriculture-based products. And here we started to collaborate with them and extract natural products and then de-orphanize their targets to figure out exactly how they work. A lot of the hesitation using some of the Ayurvedic medicine or the Chinese medicines in the Western culture is because we don't know how they work. If we can figure out, I think the adoption will be much more easier. So that's one thing we're doing. Now, we talk about pain medicine and pain management, but the first thing you think of is medicine. But no, you can do other things. Of course, yoga, laughter medicine, and all these things can be done. I'll give you an example of one thing we did in Arizona. So we're sitting in a room with a moderate amount of light. Light, as you can see, is made up of different colors. So what we did here in this particular story is to break light into different spectrum. So green light, blue light, red light, and so forth. For example, blue light has been used in Scandinavian countries to treat depression because they don't have a lot of light. There's no sun for months on end, so people get depressed. So it's been used. So we said, why not? Could it be useful for analgesia, for pain medicine? So we did. So simple experiments: take a rat, put it in a cage, line the cage with a little bit of green light, and let it expose there for 3 hours a day for 5 days. And I'm talking about 100th the light of this. This is probably 10,000 lux. We did like 10 lux or 20 lux. Well, lo and behold, after 5 days, the mice or rats were much more resistant to them being prodded by a source of heat. So whereas normally they would withstand 10 seconds, now after the green light, they would withstand 20 seconds. Fantastic. So it works to the visual system, but it's in rats, so who cares? We have to work somehow to look at the studies first. So we translated this into humans. So Mohab Ibrahim, a physician scientist who I mentored, he took migraineurs and fibromyalgia patients, about 12 or 15 of each, gave them green light to take home. Only instructed them that 1 hour a day minimum, you have the light at your arm's length and in your visual cue, and just look at it. You could be reading, you could be cooking, you just have it around. Well, guess what? After 10 weeks, they came back, reported everything. 70% reduction in pain for both groups, 70% reduction in betterment in sleep quality. Their other medicines that were taking 70% or 75% reduction. So simply something as pragmatic as green light could be useful. Now we're looking to see how we can transfer this into a patentable use of green light. So we're fabricating contact lenses that could be useful. So this is one example. So I'll give you a little bit more examples of how we're doing this. So here we take natural products from desert plants. We look at how they work in animals, in cells that conduct the pain pathway. And so, for example, we can take a structure like this, which is isolated from lavender, and we can then record from cells and neurons that convey pain. So either dorsal ganglion neurons or in the spinal cord, in the pain process pathway, and we can figure out exactly what these compounds are working on. And although nature's already evolved them to be the best compounds, they can only produce by extraction. So perhaps understanding how they work, we could synthetically make them in a simpler, cheaper, faster way. But nevertheless, it's important to know how they work. So this is one thing we're doing. The other thing I just told you in great detail, this is the real data that I was talking about. Both migraine and fibromyalgia people had significant improvement in the quality of life indices and scores, and basically that would be a very easy translation to any country where we could adopt and start a big trial. This was only about 15 people each. Now we're looking to start a trial with thousands of people. I'll tell you, a lot of the work is very heavy into biochemical and looking at protein-protein interaction. Because let's face it, pain has to start somewhere, and some of the proteins that start pain are these ion channels I was telling you about. So here we look at using information processing, using pathway mapping through protein interaction. We develop little unique peptide signatures, and then we use empirical computational modeling peptide to screen millions and millions of compounds, perhaps 27, 28 million compounds, and come up with a small molecule that can do the same thing as a small peptide could. And here's an example of a peptide we identified. It's a 15 amino acid peptide. We mutated it. We did various things to it, and eventually came up with a small molecule that could essentially be important for pain relievers. Thank you. I'll tell you one more example.
I think
yes.
Founder president is here.
Oh, wonderful! Great to see.
I am from the very beginning there. I heard you. There had VC Dasa, and I was from the very first minute with you all. But just appeared on the picture because I was in another meeting. Please go ahead.
Sir, would you like to make some remarks?
Please, let me listen, Rajesh Khanna ji, and then some comments, question answers. I'll be there because this is very. You go ahead. I'll be there.
Okay, thank you, sir.
Thank you so much. Right. Okay. So these days everything is all about computational and AI and so forth. So we here have used computational approaches using crystallography, using high throughput screening to come up with small molecules that could also be useful for pain. And what we're targeting are important ion channels, proteins that are important in the pain pathway. So here's an example where we took a pocket that we thought would be important for drug discovery, for drug targeting, and docked only 500,000 compounds and came up with a compound that was indeed selective for that particular channel. And this compound is now being funded by the National Institute of Health for one of my companies to take it forward to do extensive SAR, to do medicinal chemistry, to improve and bring it to the clinical stage. Okay, I will take a few minutes to go detailed into this particular story because it's very exciting. So this is a particular channel called the NAV 1.7. It's a sodium channel that obviously passes sodium. It's called NAV 1.7 because it's voltage dependent. Voltage changes between the membrane, opens, and the sodium comes in. And it's important because this channel is present in the right location in your pain highway, in the pain signaling system, for it to be important. And I'll tell you very briefly, so we are all on the same page. Why is this channel important? It's important because, as you all know, the language of communication between neurons is an action potential. That's how every neuron communicates. Well, this channel sets the threshold with which the action potential fires. So that's critical. Well, that's good. That's all the physiology of it. But why is this channel important for pain? Well, about 25 years ago, it was discovered in few Pakistani families that mutation in some regions of this protein caused excruciating amounts of pain in these people. So these people, for example, if I would go from this room to maybe outside, which is a little bit warmer, maybe a degree warmer, these people would have flare-ups of basically red hands, feet, rectal area, complete red, and not just discoloration, excruciating pain associated with that. So this was because of mutations in this channel. Fast forward 20 years, in a population in England, it was found, the opposite phenotype was found. People were found with mutations that made them feel no pain. So obviously, when you're a drug company, big CEO drug company, you're looking at what target we should go after. Here is a protein where you mutate one side or the other, you have gain of function or loss of function. This is the right target. So they all put money into it. Billions of dollars went into it. So that's one reason, because of the clinical phenotyping and the clinical validation. But also, it's present in the right places at the right time. It's present in the periphery, in the peripheral terminals, where you can sense pain. It's present in the cells, nociceptors, which are the pain-sensing cells. And it's also present along the pain pathway in the spinal cord. So it's in the right location at the right time. But it has revealed nothing. Probably billions of dollars have gone into this and it's still not given any real compound that's come to the clinical stage. So what we thought, we'd step back, let's not do everything what everybody else has done and repeat the same mistake. Because all they were doing is trying to block the pathway for pain. But that's bad because you and I need pain to live. So we thought perhaps we should try a different approach. So the approach we took is to understand the molecular conversation that the protein was having so that we could tweak that conversation without touching the protein. That's really the general sort of simplified concept. And so we came up with from the needle of the haystack of proteins, we came up with a particular protein. The name is not important. It's called CRIM2. It's an axonal guidance protein. Neuroscientists will appreciate that. It's an axonal guidance protein, but my lab over the last 10, 15 years has uncovered a new role for these proteins in pain regulation by regulating this very important channel. And I'll show you in the next few slides how that happens. So essentially, this protein CRIM2, whatever it's called, it's an axonal guidance protein. What we basically discovered is if you tweak this protein by just one amino acid mutation, it now no longer gets modified by a particular modification called SUMO. Proteins are phosphorylated, proteins are ubiquitinated. This is just another one of those modifications, SUMO. If you prevent this SUMOlation, you now don't allow this protein to interact with the sodium channel. And what happens is the sodium channel is less on the surface and less sodium comes in, which is perfect because that's what's needed to calm down your hyperexcitable system. So in order to prove this, we made a mouse. We took CRISPR-Cas editing approach, made a mouse that lacked SUMOlation of CRIM2. That's all we're changing. We're not touching the channel. And here I'm showing you recordings. So these are electrophysiology recordings. We're taking cells, recording from them individually. And you see these beautiful traces, downwards traces that close. These are sodium traces. From these mice that have this mutation in CRIM2, just a single mutation in an unrelated protein, interacting unrelated protein, now you see it's completely suppressed. And it's because it doesn't get to the membrane. Here, it's on the membrane in the wild type, but it doesn't get to the membrane on the mutant. So why is this important? So with this phenotype, channels are less, we should expect less pain. Well, this was a dramatic phenotype. It was amazing. This result was probably one of the best results we've had in probably 5-10 years. So here what we do, so in order to study pain, you have to pain in an animal. Either you do it through chemotherapy-induced neuropathy, you put in drugs to manifest pain in the animals, or you cut some segments of parts of the nerves. So in this case, we're cutting a part of the nerve, the tibial and sural nerve, and we leave one nerve intact. And what happens is over a week or 2 weeks, the animal becomes hypersensitive to touch. So you have these Montfray filaments, hair-like filaments. You touch, animal's okay, it won't react. But if it's in pain, even a gentle touch, it will evoke a response. So obviously, this is not, it's just a surrogate for pain, right? They cannot articulate like you and I can, but this is the only way we can test pain. So here, most importantly, we now take the wild type animals, injure them, they develop pain. So in this kind of a graph, this is no pain, and down here is a lot of pain. So wild type animals, both females and males, develop a lot of pain and they stay in pain. But the animals that had this single mutation in CRIM2 to delete the simulation or incapable of simulation, now you can see first, they do not develop the amount of pain that these animals do, but very quickly they recover, and by day 50, by day 35 or so, they are as if they had no pain. Remember, they've had the same injury as these guys. So this just goes to show you that this, just looking at the pathway, understanding the biology, but not touching the channel, we could still control pain. So great, that's all genetics and everything, but let's talk about small molecules. We're here to develop molecules. And so this is the approach we took. I formed a company called Regulonics, regulators of ion channels. Marry the two, and that's Regulonics. So Regulonics, his approach is to indirectly regulate NAV1. This channel and to prevent pain, with the idea that if we understand the conversation of the channel and can convert it into this phenotype, we have less pain. And with this, we use an in silico approach, because remember, we're trying to target CRIM2. So with my wife's group, we solved the crystal structure of CRIM2. Then we took an in silico approach, docked many compounds to the pocket where simulation happens. And you can see lots of compounds come in two sites, and we chose one and we started to develop it. And I'll tell you very quickly, and it's a busy slide, but the point is the compound is selective. So one thing I didn't tell you, 1.7 is the channel we're working on, but there are 8 other members, so 8 other cousins. We don't want to block them. 1.5 is in your heart. If you block that, hasta la vista. It's no good. You solve pain, but you also solve, well, you're dead. So we had to show that it was specific. So here we are recording again. So these are just from regular rat neurons. So these are sodium currents, regular. In control, we add our compound called 194. You see a beautiful block. You have there's a telltale toxin, a fingerprint toxin that we can use to tell what's 1.7. You can see that blocks it. But if you combine these two, 194 and this guy right here, you see this blue curve, it's the same. So what it means is that all of this block is because of specific action, indirect action on 1.7. And we profiled all the other channels. Nothing else was affected. Amazingly, it works in rat neurons, in mouse neurons, in pig neurons, but also in human neurons, which is very important. If you're to drive translation into humans, you need to de-risk your code, right? So that's very critical. And just to close the loop, because we had access to the mouse we had made previously that has no simulation of CRIM2. Now in this mice, if you take neurons and put the compound, nothing should happen. And that's exactly what happened, nothing. So you see these two are the same. So that was very nice. So closes the loop that it's a mechanism dependent on this. So in the last couple of slides, I'll tell you, because I'm studying chronic pain, we can inflict models of chronic pain and then use this compound like we did for the genetic mouse that we did. So here we take the same compound called BPBI or 194. So here we're giving chemotherapy to the animals. We inject them with oxaliplatin or platinum-based compound, 4 injections, and after about 10 or 12 days, the animals become hypersensitive. They're in pain. a lot of humans, 70 to 80% of humans that are on any of these drugs will develop similar chemotherapy-induced peripheral neuropathy conditions. Very, very painful. And in these animals, when we give our compound, you can see it's not robust, but it's about a little bit of block orally, and we're trying to make this better now, and I'll show you some examples of that. And also in nerve injury-induced pain, we're able to see oral rescue, orally based delivery that rescues, gives pain relief. Now, we can also test them. So there are issues with pain testing because you're testing by evoking. You're always evoking something. That's not really real. When you go to a physician, they're not doing that to you, right? For the most part, hopefully. And so there are other ways to test pain. And this is an example of a conflict avoidance assay where the animal is giving a choice between the light or the stage. The animals don't like to be in the light. They want to be in the dark. So you train them, and the floor in between is plain, is smooth. And on the third or fourth day, you now give them the same paradigm, but the floor is raised. It's got little needles coming out. Now you injure the animal, do the pain models, put them in the drug, put them in the chambers, and if they have pain relief from your compound, like our compound, they will go to the other side. And that's exactly what they did. So the compound works. The other thing is to remember, we started off by saying opioid addiction. We want to make sure the compound is not addictive. So we tested it in models of place preference. These are very old Pavlovian conditioning kind of models where you give the animal a drug in the chamber and see if the animal seeks it. It didn't. And then finally, we tested models of withdrawal. Anybody who's addicted will go through withdrawal symptoms. You can precipitate that in animals by jumping. So you give them morphine, they get addicted, you withdraw the morphine and then give them naloxone, and the animals manifest by jumping behaviors as an index or a proxy of withdrawal. And in neither case, our compound affected any of that. And finally, I'll tell you, of course, when you develop a drug, you have to make sure it doesn't have any side effects. So we looked at that. It doesn't have any effect on locomotion. It doesn't have any effect on exploration or anxiety, on despair, and anosmia. A lot of the things you've been noticing and hearing about in COVID is causing smell, olfactory anosmia. The people who have a loss of 1.7, congenital loss of 1.7, they're anosmic. So we have to make sure that our compound was not doing that. You want to develop a drug, but make sure you don't influence that so it doesn't do anything there. So I'll end there. We have a worldwide patent on this. We have several patents on 194 and related family of compounds for chronic neuropathic pain, for acute pain, for post-surgical pain. And what I've told you in a very whirlwind, small talk is that we began by trying to understand and avoid targeting this channel. But we did so by trying to understand molecularly how we could use knowledge of some of the key interactions to come up with a strategy that would still be able to regulate and affect the channel so we have less sodium coming in. We did that using computational approaches. We did structural biology. We then took and developed those molecules, tested them in vitro across multiple species, from neurons from multiple species. We then showed that the compound orally was effective in relieving pain, and also showed it was not addictive. The one thing I didn't tell you, which is equally important, is that even if this compound is not the best compound when it comes to market, what if we could combine it with a little bit of morphine? Let's face it, in our lifetimes, morphine is not going to be banned. It works for many things. So what if we could use a subtherapeutic dose of morphine with our compound, which is what we did. Here we have an example of subtherapeutic dose of morphine does nothing. Our compound with very low level does nothing. If you combine the two, boom, you have an effect. So here's another idea of how to take things forward, but using some of the information that's already out there, even if your drug is not as good as it might be. Okay, in the very last slide, if you permit me, I will tell you one little story that we fell backwards into. It's COVID, it's 2019, 2020, COVID hits. Everybody's, the labs are shutting down and we're kind of not working in the labs. So everybody's reading a lot of literature. And it so happened that I was telling you this protein I called CRIMP. that we discovered and we've been working on. In about 2020, there were 2 science papers published that showed that a protein. So most of us know that SARS-CoV, the virus, enters through which protein? ACE, right? ACE. But that's not the only gateway. There's another door. So there's 2 doors here. This is ACE. That door right there is neuropilin. There's another protein that was discovered that allows the virus to enter. This is a protein called neuropilin. Well, guess what? Neuropilin sits upstream of none other than CRIM2. So we were pretty excited about this. So we jumped into it and we published one of the first papers linking ion channels and showed that SARS-CoV-2 blunts pain. I'm going to repeat because that sounds very counterintuitive. This virus in the initial stages in asymptomatic people is actually giving you analgesia, which is very interesting. And if you think about it intuitively, that makes sense. As a virus, if you want to spread as far and wide, what great an advantage to give you the sense of pain relief, right? So I'll end with this slide. We mapped a whole pathway and identified neuropilin as a new player in chronic pain. And now we have several awards from the National Institute of Health with the company and also for my lab at NYU to look at and developing small molecule inhibitors of the neuropilin pathway, which are not only useful for pain, but of course are going to be useful for blocking viral entry. So here's an example of taking drug discovery for pain, but taking it further as well. All right, well, with that, I'm going to stop and thank some of the people that work with my lab, in my lab, my long-standing collaborator, my wife, who's a crystallographer. It's good to have this in the family so you can bounce ideas off. But I do appreciate the opportunity to spend some time. This could be a beginning of a beautiful relationship between NYU and Amity, and I see the power that you have. And again, I'm going to repeat with what I began with. I think you have power in all facets and then some, but pain is lacking. So I think we need to have an initiative to start a pain center. And I don't mean a clinical center, I mean a research-based center. It's critical as the population ages, as we have more and more diseases, more COVID-like conditions. I think pain is going to be an important facet of every single one of them. So I think the more research we can dole out at pain, I think the better it will be for humanity. So with that, I thank you very much.
Thank you, Dr. Khanna, for a wonderful talk and just taking all of us through your research. And you have very elaborately and very simply, narrated the rationale and the approach of your research. And you also highlighted the synergistic effect of your molecule with morphine. I think a wonderful idea. And you also narrated the role of CRIMP in SARS-CoV-2. I think you have many, many wonderful avenues open. And I think we will, can you take a few questions?
Absolutely.
Now the floor is open. Any questions?
Please tell your name and institution, please. Your name and institution. Before you ask question, your name and institution, please.
Hi, I'm Dr. Malika, and I'm from Amity Institute of Neurosciences. And my question was, because you showed CRMP2, and the last slide, I saw it was interacting with calcium channel there. So when you made that drug, was there some level of non-specificity you would assume to happen with by interaction with other kind of partners?
Thank you. That's a great question. Can you all hear me? Can you hear me, Dr. Chauhan?
Yes, please.
So that's a great question. So you're right. Specificity is a big thing. Side effects. So the amazing thing about CRIM2 is that it is a pleiotropic function protein, but the interactions are driven by the modifications that are on it. So it's like a light switch. So if you phosphorylate it, it talks to the calcium channel. If you sumolate it, it talks to the sodium channel. So it's a flipping of switches on or off that makes it specific for one pathway or the other. So great, you observed it. There was no effect on, for the SARS-CoV-2, yes, it affects, but 194 only affects the simulation of CRIM2 to talk to the sodium channel. It doesn't touch calcium. That's a great question. Yeah.
Thank you, thank you. I have one more question. In the first slide, you showed that green light, it's affecting the migraine patients, right? So what is the mechanism you would think about?
That's a great question. So when I first published this, people would look at me and like, how does that work? That doesn't make any sense. So the mechanism is very convoluted. It's recently been mapped out that the light has to enter to the obviously visual system. It's the lateral geniculate nucleus that's important in this whole process. So if you ablate it or somehow perturb its development, you do not see the effect of green light. So it's through the visual system acting directly on the lateral geniculate nucleus. And this has been shown using optogenetics and deletion and activation studies. So this has really been recently mapped out. It doesn't work on colorblind people, just to let you know, but we're still looking into the mechanisms of how it's working. And so colorblind people, definitely not, but it seems to work with migraine. Even though people with migraine have a lot of oral-like conditions, they're very sensitive to light, it still works. Yeah. Thank you.
Dr. Khanna, Malika Chatterjee is one of the most brilliant scientists in AINN, and Dr. Malik is heading it. And then our colleague Rachna and also colleague Vijay Kumar and also Vijay Kumar, Malika and Rachna, and they are all in this institution. I'm telling their name because all are related, but this institution will be your focal institution to work on pain. MIT is for neuroscience and neuropsychology, and Dr. Malik is a great experienced scientist I brought from JNU. But all those present here, my brilliant, Malika has studied abroad and in various institutions. So they all, I have named a brilliant scientist in this area. And therefore, you see the questions she asked. If you appreciated the question, it means my opinion about her is right. So thank you. Next question, please.
Absolutely.
Rajesh. Yeah, nice talk indeed. No, so basically, when you talk of the pain, you cannot keep the immune system away, correct? So this neuroimmunological network, largely sense the onset of the pain feeling, correct? Yeah. So considering your work on the CRAM2, my question is that is there any interaction of the CRAM2 with chorigenic receptors in the Piscean granules, because Piscean granules are the sensor which sense the pain, and they actually lead to the oscillation of the calcium and sodium. So is there any ABC transporter or sodium pump associated with the cramp cytoplasmic domain, or is there any interaction of this cramp with the sphingolipid 1-phosphate, sphingosine 1-phosphate receptor, which is GPCR coupled?
Right.
So this is more relevant for the neuropathic pain. Yeah.
Great question. Thank you so much. So indeed, because of the fact that this is a big protein, cytosolic, and it does interact with other proteins. So another protein that interacts with is the sodium calcium exchanger. There is some connection to the S1P receptor as well as the NMDA receptor. So there are some receptors that this has been shown to be involved in. Now, as far as your immune interaction question, that's very exciting because that is something that is more and more being appreciated, that the immune system is critical for both the chronification of pain, but also the resolution of pain. And really the resolution is what we focus on for inflammation, right? So it's very important.
Yeah, considering the connection of the cramp with the calcium, I can only see that it might have an influence on the mast cells migration and the DC migration across the histocytes.
Yeah, in fact, that's another great observation. So in fact, CRIM2 it's mostly only present in neurons, but the only other place it's present in outside the neurons is in lymphocytes. What is it doing there? So in a lymphocyte, these are spherical cells, mostly round spherical cells. It's present at the edges of the cell because when the cells have to extravasate from the microvessels. What it does is it deforms the cells, sort of collapses them a little bit, collapses the response mediator, collapses them so that they can squeeze out.
That's what claims.
Exactly. And it's amazing to see because in cases of immune encephalitis, there are two kinds of T cells, one with super high levels of CRIM2 and one with low. And in people that have this disease, they have a very high level. So this is very important.
Yeah.
Very nice talk. Thank you so much. So, CRIM2. So there might be other.
Please identify.
Yeah, I'm Pallavi. I'm from Molecular Medicine Department under Professor B.C. Dasa. So there might be other CRIMs apart from CRIM2, and if they have similar function, so would those be also targetable? And the other question I have is that UBC9 you talked about. So I mean there are various of studies with doing inhibitors for DUBs and conjugating enzymes. So what's your thought on that?
So first, Dr. Chauhan, you're absolutely right. These folks are brilliant. They know everything about it. It's amazing. Congratulations to you for setting up this. So UBC9 is a very important target. UBC9 targets many other substrates. So the first thing we did in our screen when we identified 194 is to do what's called STD-NMR, which is to look at the interaction of a small molecule with various proteins. So of course it bound to CRIM2. It didn't bind to CRIM5, and it didn't bind to UBC9 because UBC9 is embryonic lethal. So we didn't want to touch it. So that was our first cutoff. If you touched UBC9, it was garbage. With respect to your first question about other CRIMs, that's an excellent question. Absolutely. So there are 5 members of the CRIM family. CRIM2 controls 1,7 and in other ways, as you were asking, calcium channels through a different modification. The other members are not known in the context of their regulatory capacity. The other members are mostly early on in development, and then they subside. CRIM2 is the only one that is coming on and stays on during development. So I think, although those other CRIMs may be important developmentally, I think by the time development's finished, they are no longer consequential. So those are great questions. Thank you.
Thank you.
I have one question. So this is Dr. Vijay from AINN. So basically this concept that nothing works in isolation. So you have clearly appreciated this thing and this thing. So I am just curious about knowing what are the downstream signaling molecules or the partners of CRIM2 that could also like activate the sodium channel.
Excellent question again. So nothing is in isolation.
Yes, his VC has come. Great. Welcome, Vice Chancellor. Welcome.
Madam, we welcome you. VC, madam.
I told without her, the event will not finish.
So remind me a question again.
So I am looking for some
downstream effectors. Yeah, so we're looking at that. So by happenstance, we looked at and found neuropilin as an upstream effector. So the downstream effectors are more in the realm of some of the kinases, some of the enzymes that modify the protein, and the interactions of CRIM2 are sort of only being found out now. But they are mostly in the realm of neurodevelopment and axonal guidance, because this is what the protein was discovered in initially. It was a axonal guidance protein.
So CRIM2 is also sensitive to the mutated sodium channel. That is NAV1.
That's a great question. We haven't tested that. So you're asking whether the mutant would be affected. No, we have not tested that. But we have tested our compound 194 on some of the mutant channels in cells, and it also corrects that mutational deficit. So it can rescue that.
Thank you.
Yes. I was wondering. I'm sorry.
I just So we know that sexual dimorphism exists in perception of pain, and males and females perceive pain differently.
So there are studies which say the estrogen receptor is involved, but with regard to the ion channels, if you could throw some light, like how is this distinction created? Is it only because of the receptors or something else? Or the ion channels behave differently? How are these mechanisms working?
But she asked it.
Male and female, which I showed in the Nature paper in my PhD thesis from BHU. So this was my question also, that there is a difference in the male and female, the perceptions of the pain. So what could be the reason? We know some of the things that there is a hormonal difference, but other than that, what is that it? Because anything pain or any problem, you take the steroids. So steroids is highly expressed in women, but then what is it in the?
Sure. Another excellent question. You're right. You guys are on the cusp of everything that's very relevant. I'm not just saying that. Sexual dimorphism is one of the most important questions in pain research right now. And we're finding more and more that absolutely there's sexual dimorphism in pain. And the reason for that is not estrogen or any of the hormones. It may have some factor, some contribution, but really it comes down to cells. It comes down to, in males, the microglia are responsible for pain genesis and pain quantification. In females, it's T cells. So there's a different cellular phenotype and origin that contributes to this. Now, the perception and all those other factors, of course, are different in different cultures and so forth. But if you're looking for a molecular origin, it is males, microglia, females, T cells. Yeah.
Seema, you have submitted a project, Dr. Seema. BC Das, you told me Dr. Seema has submitted a project on pain. What is that, Seema? I was talking to the BC Das that you both have submitted some project. What is that?
Yes, sir. So we had proposed to design a conjugate which would target or alleviate pain in cancer. So that was what we were doing, and these are natural products, and we've used self-emulative linkers to connect the two natural products, and we wanted to deliver it in the cell and then look at the synergistic activity.
That's a good idea. Yeah, that's great.
So
any idea, Dr. Rajesh, any advice for this project? They have submitted anything which can make this project still better, research project?
Well, I mean, that sounds like a great idea. I think my only advice would be to figure if you could somehow design them to only target the cancer cells. Then that's the $1 million question, right?
Yeah, so that's why we used ER beta agonist as one of the targeting agents so that it goes to the cancer cell. So we used that as a ligand, which is also an agonist. It targets the ER beta and it will be delivered in the cancer cell because.
Fantastic.
So that is what we had designed.
What about nanoparticles, using sort of a nanoparticle type of approach?
Yes, so that would be the next part of the study. Once we have the conjugate and we can establish proof of concept that they act synergistically. So once we have that, then we can encapsulate them and.
Wonderful. Great. See, there you go. They're already doing it.
Hello, sir. I have one question.
Back there.
Yeah. So I'm Dr. Rachna from Department of Neurosciences. So you have spoken about the chronic pain. So I was wondering, because there are different types of pain. We can call it a heat pain or a cold pain, neuropathic pain and all. So can we talk of sodium ion channel dependent, voltage gated sodium ion channel dependent mechanism being present in the different types of the pains? Can we consider it as a generalized mechanism?
Yes, it can be, because as I told you, it's 1.7 kickstarts the action potential. So if you don't have that initial part, you don't get the action potential. 1/8 carries the massive part of the upstroke, but without that little piece, you never get the firing happen. So I would say absolutely. I think there are some pains, for example, visceral pain, that this channel has no role. But for the others, I'm fairly confident that it's important for all of them, including very important for cancer pain.
Right. So one more thing, you have spoken about the simulation-based mechanism, right? So here can we rule out the important role of other type of modifications like ubiquitin annihilation and others, or their involvement being present in any such kind of mechanisms?
It's a very exciting question. Very broad question because ubiquitination happens on a lot of different proteins.
Right.
So I don't know. This is a very specific modification. As you know, ubiquitination and simulation happens on many proteins.
Yes.
The challenge is to find something specific. And the way we did it is to take structural information and use that to guide the discovery of simulation inhibitors of CRIM2. These inhibitors are not of all simulation, just of CRIM2. People have tried to discover sumo agnostic substrate inhibitors. Disastrous. They're very non-specific. They block simulation of everything. That's not useful. So I'm not sure if that would be the way to go. I think you need to find and sort of selectively thread the needle as to find a specific simulation or specific ubiquitination.
Right.
Ubiquitination might be a little bit harder because it's a lot more prevalent. Yeah.
Thank you.
Thank you.
I have a little one more question, very brief. Just I was wondering if I have understood rightly that the neuropilin A, or you are turning neuropilin B, because neuropilin is mostly involved in the cancer progressions. But how that is that any relation to CRIM2 or anything?
Yeah. So thank you. Yes, exactly. Neuropilin 1 is the protein we're working with, and it was discovered in the context of cancer 25 years ago or more. So it's very well known in the context of cancer, and it's important because its ligand is VEGF, vascular endothelial growth factor. And why that is important is because that's what's one of the things that helps promote cancer. And so compounds and small molecules have been developed that actually are used in the clinic. So we're now trying to see if we could repurpose them for pain, because they have never been useful for pain until our story came out of just luck. You know, serendipity is a big part of science, right? So we just discovered neuropilin by just chance, because we were looking at CRIM2 and we knew CRIM2 and all the story behind it, and we discovered neuropilin. So I agree with you. Neuropilin is very exciting, and it has a relevance in cancer. But I think we can now try to see if it could also be used to target for pain.
It is discovered, it is working long. He has a huge contribution. You might be knowing neuropilin Dev Mukhopadhyay. He is in the US. So he is also our honorary professor from here in MIT. So he works on this neuropilin.
Wonderful. Dr. Basu.
The molecule which you have discovered, we have shown that it is much more efficient with respect to the morphine concentration, with respect to the doses is concerned. But this morphine, all these things are very temporary kind of things. So what is with respect to your molecule, how much it is with respect to the lasting is concerned? And you have shown very clearly that in case of morphine, patient are addicted, but in this new molecule, patients are not addicted. That's a very important part of it. But what is the effect? How long it is staying? Number one. Number two question is that you have also shown in your last slide that the COVID-19 blunt pain. So in these cases also, because it is, I think, affecting the olfactory nerve. So how many means what is that lasting time, that how many days or how many months the COVID patient, that depends on the extent of the disease, or what are the factors?
Sure. So I'll answer the second question first. So COVID-19 pain relief, it only is transient. So it's only in the asymptomatic or pre-symptomatic stages. So it's very short, 7-10 days. But so ever since we published that paper, I got reports, unsolicited reports to my email from various people across the globe that said, I'm a chronic pain patient in South Africa. I have pain for the last 20 years, and I contracted COVID. Pain is gone. And then it would email me back 3 months later, my pain is still gone, and so forth. And some other people wrote to me and said, when they're chronic pain patients, they got their first vaccine or the second vaccine, and their pain was gone for a week to a month, in some cases forever. I mean, these are people who are taking chronic pain meds for decades. So something is happening. We don't know what it is right now, but something is clearly happening. So the time window is very labile. I don't know how to answer that question, but there's something here that we're trying to figure out. As far as your first question, morphine's effect is very short lasting, and that's why you need to take more. But then it becomes tolerant, so you need to take twice as much, and then 4 times as much. And then if you take too much, respiratory depression, and you die. Our compound is, the compound I showed you, 194, has about a 4 to 6 hour window of relief. But here's the catch. In the interest of time, I didn't really show you. So 194 was taken and awarded into the program by the NIH. It's a special institute called the National Center for Advancing Translational Studies. They took our compound and put all of their team of 30 medicinal chemists on it. They made 400 additional compounds. And our 194, which was published in Science a year ago, has like a 20-minute half-life. Their best compound, their best compound, because we own it still, has a half-life of 22 hours. So the point is that, yes, our compound, with all the stuff we had, we published because it was our best compound, a small lab, we could only do so much. But with the power of the NIH, with power of big, like it's almost like a small pharma, we've been able to increase the efficiency of the compound. And now with a 22-hour half-life, we give the compound once and we have pain relief for 3 to 4 months. So we're getting there. But again, if you're a drug company, you don't want to develop a compound that gives you pain relief from one oral dose for 3 months. You'll be broke, right? So a lot of the people that are in positions of power say, oh, that's not a good compound. And I'm saying that's the best compound. No, it's not a good compound. But you see what? This is so.
Just last question. If it can be converted to be, I'm talking about your compound, it can be converted to be a sustainable release to the delivery system or stimuli responsive. Do you think they will give you a much better result?
Yeah, so I showed you an example of that in the Allzat pump. So this is a pump that we basically make an implant in the back of the animal, and it's like a little capsule. We put it in, and it delivers constant. And you saw that the dose lasted for 15 days because slow release, exactly. So we could do that, but they have made a compound that's 22 hours. That itself could be also made into something like that. But you're right. Our compound still could be tweaked. It is fully patented worldwide, and the compounds that NCATS from the NIH has developed, we co-own the patents. And so I'm not sure where to go. Right now, we're at the stage where we're partnering with investors to see which compound they could take to first in human in healthy volunteers. Yeah.
Founder sir, I think we will close for the questions. Sir, your comments, your remarks to all of us.
So, taking things of your order, Rajesh, I must tell you, in this campus, we have in a year at least 1,500 events. And this today's event, your speech, interaction with my worthy faculty members, researchers, is among the most important event today. I tell you because in my eyes, pain is a very, very important area. And to all my faculty members, Vice Chancellor, you must be knowing that quite some time back, I got diarrhea-induced pain and was 5-6 days in Apollo. And Rajesh will understand more what is diarrhea-induced pain. And the best doctors of the country were treating me, but nobody could tell me the reason, what is this diarrhea-induced pain. And Dr. Khanna from Apollo is one of the great leader in the pain area. So today, your lecture would lead to a big history, not only in India, but globally, because I feel, Rajesh, that the Indian systems of medicine, together with the Western system of medicine, can do wonders. only Western system or only Indian system may not be that successful. Therefore, your appreciation for my researchers. I must tell to my scientists, one day a journalist asked me interview, what gives you, sir, the biggest pleasure? I told, I get the biggest pleasure when my scientists are appreciated by some expert. What Rajesh you did, but it also showed your deep knowledge, deep experience in all these areas. So you could very worthily reply each question to the satisfaction of my scientist. Dr. A.K. Singh, generally when you go in a cricket ground, the player takes few minutes or half an hour to get warm, but you have started hitting sixers from the very beginning day. Calling Rajesh ji is a big sixer. I think this is your first invited lecture. I must appreciate you.
Thank you, sir. Thank
Dr. B.C. Das, that such a great scientist of international repute could introduce amity I was listening very carefully how with competence you introduce everything about amity, which even I don't know myself. Dr. Shukla is developing so fast the institution, researchers, and statement. Just just a minute. This what has happened? Just a minute. This I am not on this thing. Right. And Dr. Bisidas, I was really surprised. And the third great thing is that all sitting over here from different institutions, I know about these institutions, I think it was a very right selection, those who all will help me in making a center for research. Now, at this stage, Rajesh, can I ask you, what should be the right name? Because I want to do a lot of work nationally and internationally and want to be, pardon me, it is not an ego, pardon me, it is my mission to be the biggest in pain research in the world. I believe we can, if a person like you guide us. So what should be the name of this center? Center for Pain Research or what?
The Johan Center for Pain Research?
Hello, sorry, I couldn't get.
So I was joking, but I was serious. I was going to say, I was proposing Johan Center for Pain Research.
I got one, so I'm going to make one center for sleep, one center for. So Johan's name should be away because should we make a foundation for pain research? Is it okay, pain research, or any other addition to this?
Pain sciences is a good idea. I like that a lot. I think one thing we talk about pain, but pain goes hand in hand with addiction.
Right.
So I think, in Arizona, when we set up a center before I left to New York, we had set up a center called CPAC, which is the Center for Pain and Addiction. So, or the Comprehensive Pain and Addiction Center, CPAC. So something like that would be a good, because it's all encompassing. Because if you look at pain is huge, but you cannot ignore the other half of it, which is addiction. So perhaps putting that in is also important, but that's up to you.
Addiction from what?
Addiction of pain, like pain therapeutics. People get addicted to some of the morphine or some of the pain drugs, right? So that's a very important aspect of pain management, is to stop that from happening. So I think if that could be a part of the center, addiction is a very strong field, and I think it's very important. Addiction can be from many different sources, opioids to substance abuse to alcohol to various things.
Right. A.K. Singh Sahab, you told that in which DRDO lab pain is tactile? Is it in mass or DPAS? Which is the DRDO? Because you told me today DRDO has make a DRDO armament research center or something. What is that? DRDO armament.
Visit of Dr. Rajesh Khanna to MIT stems from the research on pain being carried out at Institute of Nuclear Medicine and Allied Sciences.
Okay.
There, we used to run a program on combat casualty management.
Right.
When I say combat casualty management, it means the mutilations or the injury which is inflicted on the soldiers by mine blast, or gunshot or artillery shot. They lose, their extremities, means हाथ ही पूरा चला जाएगा या टांग चली जाएगी, then there is excessive bleeding. Right. And it is estimated that 60% of the such deaths are avoidable. Right. When this is avoidable, the another issue is of the pain management to those soldiers, because they are in severe pain and normal painkillers cannot manage. And if you go for opioids, then they put the cardiorespiratory distress, because already there had been a blood loss. So this is not the right option. So there had been lot of efforts to find a molecule which relieves the pain but does not put the cardiorespiratory distress. So one molecule which is this is used चूहों को देते हैं जिसको बेहोश करने के लिए भी मेरे दिमाग में आएगा. That was used as an alternative. So that because that does not put the person on the cardiorespiratory distress. So from there, I think we were publishing, and Dr. Rajesh Khanna looked at those publications, and then he came in touch with my colleague, Dr. Kailash Manda, who is expert in developing animal models. My last question was that, he has shown a wonderful research, but still, basic. How do we make a assessment or pain grading animal model? That would be a huge problem at INMAS at that time, and we could not find out. So, sir, that is the genesis of this, and I think Dr. Khanna is here. Abhi, it is not even the end of the beginning, and I think with the interest of all the faculty and the way he has explained and showed interest, I think the things will go. I think we will nurture a big and very strong relations with him, with his institute for.
Right. Gopal Bhushan ji, you please make a bridge with DRDO together with AK Singh, because everybody in your long years there. Rajesh ji, which country is advanced in the pain matters out of the whole world? If you can name me 5 countries which are working very intensely on the pain, which are these countries? Because I want to read 100 papers myself on pain. Before I do something, I read it. I've got your papers in front of me. I could not go through it. So which are the main countries?
I would say the US is definitely one of them. US, UK, Germany. I think China is making some inroads into it. I'm going to say that that's 4, right? Germany, UK, US, China, and I think the other countries that would really be doing a lot of good pain research is perhaps Canada. Okay. My adopted country of birth, after birth. So these are the 5 countries. I wish I could say India is in those 5, but I think you guys need to make that happen.
In next.
Yes.
There are so many ways, therapeutic, yoga and all. Then there is allopathic, there is homeopathic, there is Ayurveda. So which area you trust? Should it be a combination of these areas? Because I may tell you, I'm a great fan of homeopathy. There, if you have the highest pain, the Rustox 200 gives miracle. So, but you must be having other opinion. Which areas? Because all my specialists sitting over here, they are working in all these areas. So I must give a combination of areas to develop the real remedy for pain.
That's a great question. I think keeping in mind and being aware of not just drug discovery, drug-based stuff. I think Ayurvedic medicine, I think natural products is very important. So I think one aspect of what you're trying to develop could be under the guise and the umbrella of natural products, which would cover everything: homeopathic, Ayurvedic, and all sorts of medicine. Which you're right, we cannot ignore them. This has come for thousands of years. This is so much wisdom and knowledge in here that works. So I think one aspect of what you could develop would be this combination of looking at how they work. I think one of the reasons why this has not been adopted, particularly by the Western medicine, is lack of understanding of how they work. So, for example, a few years ago, when I was living in Indianapolis, I suffered from a lot of back pain that I couldn't figure out. I went to many different things, a lot of different drugs, nothing could work. Until I met a Chinese colleague who is an acupuncturist. He was a specialist. He said, if you're not afraid of needles, let me try something on you.
Right.
And okay, at this point, I'll try anything. And he put a big needle, about that big, into the brain up here. And, it looks scary. It sounds scary, but it wasn't. And I can tell you, within 30 minutes, I never had that pain again. So
Right.
the point is that I was, even as a scientist, we're all educated people, but we're afraid to try something that we don't know how it works. So if you can figure out a way to incorporate in the center ways to go into the mechanisms of how things work, I think that's critical. That gives credibility, confidence in the way things are working, and it establishes the center and really shows and showcases that, look, we discovered how this is working. So I think that's important, very much, very important.
Great. I'll tell to all my persons sitting over here, make some startups, pain related area, because these are the virgin areas where nobody has got thought so deeply, so intensively as we are thinking, as we are having a center for sleep. Sleep is half life. If you cannot sleep, you can't work. So their center we are making with Gangaram Hospital. Now make 4 or 5 startups. There's a huge money. You have know the Shivani. That project was only 1 crore. It has been now 10 crore, and that project, that concept, that research is now 50 crores. So the government is very much looking for brilliant scientists, and all those who are sitting over here can set up a startup. That will be your company. You will be the owner, and the government will fund you, and we will support you. So I think Gopal Bhushan ji, A.K. Singh Saab, Neeraj Sharma ji, please tell them all the scheme of, I want 50 startups in 3 years from MET. Yesterday there was a person from IIT Kanpur. They are having this project from the DST. They give 170 crores to them. They told Rahman, you set up with your scientist startups, will help you. So the whole world is looking for brilliant brains like you. I feel proud that no university in the country, no other lab is having so brilliant brains. Rajesh, you must have seen. I appreciate that you have understood their brilliance. So this would lead you to your readiness to help me, because you will see that with them, to your ideas, we can realize really worldwide. So my compliment to all those sitting over here. Dr. Shukla, at the last, I ask you, you're a very wise person. Is my Zanoon, is my obsession for pain right, or I am in dreams about pain? I told you how important is pain.
Honorable Founder President, thank you very much. Your question is absolutely right, because the pain is to be addressed, and research in this area, as Professor Rajesh Khanna ji has done, and many other alternative medicines, which especially Indian medicine system, as you've said, that is the combination of two. So maybe as you are already has been guiding and directing all the researchers and scientists in the university to look at the combinations of the medicines and the solutions to the pain and other areas of the research avenues. I think with the guidance of Dr. Rajesh Khanna ji, and the ideas of all the researchers and scientists which they are working. And another thing which Honorable Founder President, you have promoted in a big way, that researchers and scientists to start their own ventures. And it's a very, very unique scheme by the university that will not only promote the research, but it will also contribute to the economic development of the nation. That's what our Honorable Prime Minister is looking at, how the research academic institution, the research organization, bring the research, and which is can be converted into other technology transfer or the startups, because the startups will give the bigger dividend to the country in the form of job creations as well as economic development. So, Founder President, your initiative, your vision is which is promoting the research in big way, and you're all the time creating the new initiative that science, technology, war room, and all many things, so that the research can be promoted all the time, and that time which you are spending in the research and innovation. That's, we really extremely thankful to your honorable founder presence. Dr. Khanna, I would like to thank you very much for sparing your time. And Dr. A.K. Jain, I would like to thank you for bringing Dr. Khanna. I'm sure you said it's not the end of the beginning, it's just the beginning. And with this beginning, I think we can achieve the founder president's vision to make this as a research and innovation driven university, to contribute to the not only to the nation building but also to the humanity, and make this world a better place, to developing the synergies between the scientists from across the globe.
Great. Dr. Rajesh, last advice from you. Are you in India with your wife or you are alone this time?
I only here for a few days by myself. I wish I could bring my family with me, but just came for a few days. I told some of these guys here, Dr. Singh, I told him that, so I was born in Delhi and I migrated to Canada when I was a little kid. And this is my first visit in 36 years.
Right.
So I feel like a stranger in my own country.
Why I'm asking it, because we would like your wife to visit our campus and we will do a bonding with her so that she compels you to come more often.
Absolutely. All the family here.
If she likes us, she likes India, then she will come. Let us go to India and we'll go spend 2-3 days at the MIT campus. Sir, I tell you, I always believe in God. God has sent you to me. Without you, my ambition in pain will not be as exhaustive as today is. And I think I'll brief to Dr. Subramurthy. Dr. Subramurthy must be listening also. I'll send him the URL to all my present scientists, faculty members. You tell to Dr. A.K. Singh, he will send you the URL, because all these important lectures, I think twice on the recording. You also think. Many times when you are listening here, one or the other word you could not grasp or could not listen. So see the URL, listen it, make notes, and A.K. Singh Sam, Gopal Bhushan ji, Dr. Shukla, make a consortium from all our universities. Those who are really capable, want to devote and contribute to pain, we will have about 15, 20, 21 member consortium, so that we do very fast. We'll file the patents, we'll make the startups, we'll do the publications, and I think Rajesh ji will be kind enough to see that my people are hungry to take your help, sir. Thank you. Dr. Shukla, now we have called you for this great ceremony. If A.K. Singh says, please do the ceremony.
Sir, I think before that, let's clap for Dr. Rajesh Khanna, and let's also say thank you through clapping to our honorable founder president.
मैं तो आपके साथ हूँ सब. A.K. Singh साहब जी साइंटिस्ट को देखा, I can work 36 hours, 18 hours तो मैं करता हूँ. Each one I know, they are so brilliant from different institutions. All institutions are involved in this. So I appreciate goodwill and everybody who has invited A.K. Singh Saab. They all will be, many of them will be part of the consortium. Thank you.
So, thank you, sir. With your permission, can we go ahead?
Please.
President, sir. I think he's not able to listen. I think we are in the last leg of today's wonderful program. It had been unparalleledly enriching talk. I think I'll take liberty of saying thanks, a big thank to Dr. Rajesh Khanna, Honorable Vice Chancellor Dr. Shukla, Dr. B.C. Das, Dr. Malik, Dr. Neeraj Sharma, Dr. Gopal Bhushan, Dr. Tinku Basu, Wing Commander Shukla, and all faculty members who came here, participated, interacted, and made the program wonderful. I think the last, before the felicitation to Professor Rajesh Khanna, I think I'll say that in view of your unparalleled contributions in research, particularly in the pain management, your entrepreneurship, your inventions, and being founder of 4 companies, and your vision, with utmost humility and respect, MIT University confers the title of honorary professorship at MIT Institute of Neuropsychology and Neurosciences. Congratulations to you. I'll request Honorable Vice Chancellor to present the memento and the certificate.
Thank you so much. What an honor. Thank you all.
Thank you, madam. Thank you. Now I'll.
Thank you very much.
I think, may I request Dr. B.K. Das to felicitate Professor Khanna. And request Honorable Vice Chancellor Neeraj Sharma. Malik sir, Dr. Gopal Bhushan. You also please join.
AK Singh साहब बाद में एक बढ़िया शॉल मैडम के लिए भिजवा देना जहाँ ठहरे हुए हैं.
ठीक है सर, right sir, right sir.
We have kids with them, then some toys for the kids also.
Thank you so much. Thank you.
Founder sir, with your permission, can we close the program?
I must tell you, Rajesh ji, how important is at 5 o'clock, I was chief guest in a very big art gallery. So I told them, you all wait. I am in a very important event. And now when I go there, they inflict pain in me. I will tell to Rajesh Khanna is the culprit.
Blame me for it.
Okay, I have to rush now. Okay.
Thank you, sir. Thank you very much. Thank you to all of you. Thank you.